So this is the cheek cell extraction lab. What you're going to need for this is a microscope, some methylene blue dye, a toothpick, a slide, and a plastic cover slip. And you might want to have a paper towel handy just in case. So the first thing we're going to do for this is we're going to take 1 to 2 small drops of the methylene blue dye and place it in the center of our slide. All right, then we are going to take the toothpick and gently swab the inside of our cheek to try and get some cheek cells off. Okay, so after you've done that for a little while, you're going to take the tip of the toothpick that you had just swabbed your cheek with and stir it in the blue dye. And you can spread it out a little bit, it doesn't really matter. Just try and get all of the cheek off of your toothpick. Just put that to the side. And then what you're going to do is take the plastic cover slip and gently place it on top of the dye that you just put down. And it will expand, so if it starts getting outside of the cover slip, you're going to want to dab it with a paper towel just so that you don't have any of the dye on the outside of the slide, because you don't want it to get onto the microscope lens. So then, after you've done that, you're going to take your slide and place it on your microscope. Your microscope is on. And you're going to start it on the shortest lens, and that's called the scanner lens. So this lens will allow you to just get a full look at it and try and choose where you want to zoom in on the slide. Okay, so we were on the scanner lens, which is the shortest lens on the lens things. And this lens allowed us to get the whole view of what we were looking at under the slide. So now we are going to turn the knobs so that we get to the medium length objective lens. And this is our low power objective lens. It allows you to zoom in on what you were looking at under low power. So when you turn this, the view might go a little bit out of focus because you are getting a bigger lens. So you're going to want to look through the microscope and turn the smaller zoomy knob a little bit, just so that you can ensure that you're having a clear picture through your microscope. So now we're going to go to the high power objective lens. We were just on low power, which allowed us to get a closer view, but not quite really zoomed in. The high power will let us really close up on whatever we were looking at under the low power. So we're going to gently turn to the high power lens. Sometimes this gets a little dicey because the slide can be too close. And then you're going to want to look through the microscope again and just turn the little adjustment knob again to make sure that the view is in focus. These microscopes have what's called a parfocal, which makes it so that you never have to turn the big knobs after you've zoomed in and gotten a clear image on the low power, because it has a stabilizing device within the knobs so that you only have to turn the little knobs to ensure the clear view on the highest power. Okay, so what we were looking at in this experiment is what's called a somatic cell. Somatic cells are everywhere in our body. There are skin cells, or like tissue cells, except for our reproductive cells, which is egg and sperm. So when we stained these cells with the methylene blue dye, the really dark blue thing in the center of the squishy, clear-looking stuff around the outside is what's called the nucleus. And every single cell, except for red blood cells, have nuclei. And nuclei are like the home of all of our DNA, which is like the genetic blueprint that codes for all of the proteins and everything that makes up our body. and all of the squishy kind of clear stuff around the big blue dot in the center of the cells that we were looking at is what's called the cytoplasm, and that's the squishy, cushy stuff that cushions all of the organelles, which are the other important things inside of a cell, that kind of just keeps them in their place, holds the structure of the cell, and allows proteins and exchanges with the environment to happen.